Good morning everybody. Now we have a lecture from one of the expert and one well-known scientist, Dr. Yogesh Shouche, and it's my great pleasure to introduce Dr. Yogesh Shouche. He's a scientist G at NCCS Pune and he's also principal investigator of Microbial Culture Collection Center at Pune. Sir has received his MSc degree from Pune University and PhD from IISc Bangalore. Sir has did his postdoc from Germany and he has more than 30 years experience in research, and he has received many fellowships from ISRO, CSIR, DST, ICMR, and received many awards. Sir is a reviewer in many reputed journals and editor of Indian Journal of Microbiology and European Journal of Soil Microbiology. Sir has more than 150 publications in national and international journals and in reputed international journals like AEM, BMC Microbiology, Nucleic Acid Research, ISME, IJACM, and PLOS ONE. Main area of research in Sir lab is involved in the microbial community structure from varieties of ecosystem like human gut, marine sediment, soil, and other terrestrial ecosystem, as well as the insect gut, vector biology, and the genetic diversity of pathogenic bacteria. So it's our great pleasure that Sir is with us to share his knowledge. Any workshop related to metagenomic and microbial ecology is always be incomplete without the Sir lecture. So today's
Thank you very much, Vilas, for that very nice and crisp introduction. I'm really happy about that. See, that's the advantage of being in this field, because anywhere you go, you always come to know some students whom you know very well. So I'm also happy here that Vilas is here. She has practically worked partly with Indians. Oh, hello.
There is also one student.
Oh, okay. Sorry, I missed you. So it's really a pleasure to be here, and I had given the title of my talk as In-silico Comparative Analysis of Variable 16S rRNA gene regions. But after having a look at the other talks, I have done some more additions to that. So basically, I had divided my talk in 3 parts. The major part is going to be In-silico Comparative Analysis of 16S rRNA gene sequences, as mentioned in the title. But towards the end, I have added 2 small pieces on other applications, industrial applications of metagenomics, a quick review of that for the sake of completion, and finally I am also going to discuss again in few minutes about the human microbiome work which we have been carried out, just to give you an idea as to in India also a lot of work is being done on human microbiome and where do we stand in the worldwide scenario. So that's how I have made few additions to my talk yesterday evening. So coming to the main topic of our discussions today. So all this talk about metagenomics goes back in the history to a phenomena called the Great Plate Count Anomaly. So I mean, the basic question is, why do we study metagenomics? Why 16S rRNA? Why NGS? So I'm just trying to understand this basic question, and then this basic question originates from the old problem in microbiology of this Great Plate Count Anomaly, and the name sounds very impressive, but actually it is very simple. In simple words, what it means is that if you take any environmental sample and plate it on a petri dish to grow the organism and look it under the microscope by any means of staining, what you would find is that your microscopic field is teeming with organisms. But when you grow it on the plate, you will find a very few colonies growing on the plate. So the correspondence between what you see on the plate and what you see under the microscope is not there. You under the microscopic field you see a very large number of organisms, whereas on the plate you see only a fraction of that. So that is called as plate count anomaly, and the reason why majority of these microbes are unable to grow here, there are many reasons for that, but that's what we call as microbes that cannot be cultivated in the laboratory, uncultured bacteria, uncultivable bacteria, so on and so forth. So people were looking for means to study these microbes, and the fraction could be variable. In an ecosystem like seawater, only 0.01% could be cultivated, whereas in activated sludge, 1 to 15% could be cultivated. And then in other ecosystems there is a large variation, and this is true for any ecosystem including human gut. So people were looking for ways and means to know more about this uncultured majority, which represents majority of the microbial world, and what we know actually is only tip of this iceberg. So people were looking for ways and means to understand this uncultured microbial diversity, and the answer came in 70s by way of discovery of ribosomal RNA gene as a molecular clock. So Carl Booth is the person who proposed that the sequences of 16S rRNA gene could be used as a tool to understand microbial phylogeny. So he did not have any idea about uncultured diversity and other things those days, but he said that the 16S rRNA gene sequences could be used as a tool to understand microbial phylogeny, and that kind of laid foundations of all these microbial genome projects, human genome project, human microbiome project, earth microbiome project, what we all talk about today. It kind of laid the foundations of that, but because it gave us opportunity that looking at a 16S rRNA gene sequence, we can identify a microbe. So since it was a phylogeny tool, 16S rRNA, of course, I mean, in his work there was nothing like 16S rRNA sequencing, because those days sequencing techniques were not available. Sequencing was discovered in 1979 and this was done in 1970s. So he used a different technique called as oligonucleotide cataloging, but let us not go into the details of that. The basic principle is that 16S rRNA can be used as a molecular chronometer, and it can be used for identification and classification of bacteria. So that was the clue which was taken from Carl Booth's work, and then this led to the this science which we talk about today. So 16S rRNA then was accepted as a gold standard for microbial taxonomy. Its sequence can be used for, it was believed that this sequence can be used for microbial identification. And I mean, for those who are not very familiar with this terminology, 16S rRNA is a part of ribosome. It is associated with ribosomes as an essential component, and it is involved in interaction of mRNA and a proper binding of mRNA to ribosomes. So this was then the reason for accepting 16S rRNA gene as a molecular chronometer are very many, but the most important being that it is found in all the prokaryotes. I mean, it's present in all living organisms for that matter. Its sequence is 1500 bases long, so it gives reasonable information for statistically significant phylogenetic comparisons. As we shall see later, it has both conserved and highly variable regions, and luckily for us, these conserved and hypervariable regions are interspersed with each other, which is a very great advantage when we talk about our modern day sequencing technology. I will come back to this issue again. Then it allows us broader range of phylogenies to be covered. Again, we'll see these actual examples of this from domain to species level, and that is another advantage as we shall see later. And again, this is an issue which we are going to revisit again and again, that a huge database. Today the database stands something like 2 million sequences. Huge database is available for sequence comparisons. So and finally there is very little there are very few examples of lateral gene transfer, if any. So these are the advantages which makes 16S rRNA as a best suited molecule for phylogenetic and taxonomic identifications. This was followed by two major developments in molecular biology, helped 16S rRNA sequencing to become more popular for phylogeny as well as diversity studies, and these are, as I mentioned some time back, discovery of techniques for DNA sequencing in 1980, and initially it was a cumbersome, laborious procedure of radioactive based sequencing. And another important technique which has helped is discovery of PCR. So polymerase chain reaction, which allows amplification of a target DNA molecule. And remember, 16S rRNA gene is a mosaic of conserved and hypervariable regions. So the advantage of this is that you can make use of conserved regions. design primers which can amplify 16S rRNA gene from a large diverse group of microbes. The same primer set can amplify gene from variety of microbes, and then this can be sequenced. So both these techniques together had a great impact on microbiology as such, not just microbial diversity and ecology, but overall science of microbiology has been influenced by these. And then in order to study so-called uncultured, uncultivable microbes, the technique that was introduced in 90s was you take any ecological sample, PCR amplifies 16S rRNA genes using suitable universal primers, then you will get a PCR product which is a mixture of 16S rRNA genes from different organisms. So in order to get a sequence, you clone them in a suitable vector, and then once you get the clones, you sequence these clones, and then the sequence of individual clone would help you in identification of the corresponding organism, and thus you can arrive at the microbial community structure of a given environment. So that was called as PCR clone approach, and this was the approach which was first introduced way back in 1990s. And so when it was introduced, there were, for the sequencing, the techniques that were available was basically radioactivity based, which was time consuming, cumbersome, which was then slowly towards the middle of 90s, it was followed by capillary sequencing, which was fluorescence based. And with capillary sequencing, initially it was possible to get only 400 to 500 bases sequence in one run. Subsequently, that length was increased. At that time, the one run was typically overnight run. So 10 to 12 hours run, even on capillary sequencer. And radioactivity, as you know, it was almost a week to get a sequence. But then when when gel-based sequencing for fluorescence-based was replaced by capillary sequencing, the sequencing length increased to around 1000 bases, and the run time was reduced dramatically. So from 10 to 12 hours, run time came to 2 hours. So with that, then it was possible to study many ecosystems using this clone library approach, and its impact in microbiology could be seen in this slide. So when 1994, there were 13 divisions when the technique was just introduced. This technique of clone library approach was just introduced. There were 13 divisions, all represented by cultured organisms, as seen in blue color. Just a decade later, when the technique was a decade old, there were 80 divisions, out of which 26 had cultured organisms, and majority, 54 divisions, had organisms which were not cultivable or uncultured. So basically all the information about these organisms was the information available from cloned sequences of PCR amplified 16S rRNA gene. So that is the impact it had on, by using the techniques of either radioactive gel-based sequencing or capillary sequencing, which gives you around 1 million bases overnight. Beginning of this century, many modern technologies came into the market, and which is what this workshop is for. So I did not talk much about these techniques, but these techniques have massive power. As against 1 million bases in one day, these techniques can generate 300 gigabases sequence in one run, which would probably last for a week or so. So this great leap increase in output sequences again has had an impact on the way we do this science of microbial diversity and microbial ecology. Same time, the sequencing cost also came down. The whole aim of this technology is to generate large amount of sequence at a lesser cost. So just to get an idea as to how cheap the technologies have become, in 2001, when Human Genome Project was proposed, it was at the cost of 15,000 crore rupees and supposed to cost 11 years. Slowly, as the technologies develop, today we stand in a situation where human genome sequence could be done at the cost of meager 60,000 rupees and in less than 24 hours. And if you just want to have comparative figures, Illumina generates something like 300 GB data in one run. And if you take average human genome to be 3 GB. In one Illumina run, you can sequence 100 human genomes. If you extrapolate this calculation further, typical microbial genome is 5 billion bases. That is 5 megabases. So in a single Illumina run, where you get 300 GB data, theoretically you can sequence 60,000 microbial genomes in one run. So that's how you can generate massively huge information on sequences using these technologies, and that's why the entire way we look at this science of microbiome and microbiology has dramatically changed. But there is one fact which we should remember, is that these platforms, as you would probably know by this time, that these platforms have very short read lengths. So Illumina typically has, depending on the which chemistry you use, it generates 75 bases, or nowadays 150 bases, or 150 into 2, so at the most 300 bases sequence in a single read. Solid generates 35 to 50 per read. Ion torrent generates 300 bases sequence per read. So read length per sphere or per length is 300 for ion torrent, and FLX generates it says 1000, but currently it generates around about 600 to 700 bases sequence per read. So typically the read lengths are shorter as compared to Sanger sequencing, and then you have to use these shorter reads for 16S rRNA gene sequence based identification. So we will come to that, how we address this problem. So if you want to look at the microbial world today, there are 3 approaches based on genomics. The approach which you see towards this part, which is called as single cell genomics, I wouldn't discuss about this particular approach of single cell genomics, but when we talk about metagenomics, again the metagenomics of microbial system can broadly be divided into 2 categories. One is what is called as 16S rRNA gene PCR, where you use some specific primers of your choice and amplify a fragment of 16S rRNA gene and sequence that in order to get insight into microbial community structure of that particular ecosystem. So this is called as tagged amplicon sequencing or 16S rRNA gene PCR. The second approach is actual metagenomics. So there you do not amplify a particular gene or gene fragment, but you sequence the entire genomic content of the ecosystem. So it would contain not only 16S rRNA, but it would contain every possible gene of every bacterium present in that ecosystem. So these are the 2 major approaches, and then you can address the analyze the data by a variety of means depending on the kind of questions you are asking. Needless to say, when you have a actual metagenome run where you have sequenced the entire genomes of microbes present in the environment, you can always extract 16S rRNA gene sequences from the metagenome and derive the phylogenetic structure first, and then go into the analysis of other metabolic pathways. That definitely you can do when you have a actual metagenome run. So then there the 16S rRNA based diversity part forms a subset of your major question. But if you do 16S rRNA tagged amplicon sequencing, then you get information only about 16S rRNA based community structure, and then if you want to understand actual metabolic pathways, you have to resort to some different ways of doing that. But whichever technology you use, the fact remains, whether you are doing actual metagenomics or whether you are doing 16S rRNA tagged amplicon sequencing, the fact remains that typically you are working at shorter read lengths, anything depending on technology being used, 50 bases to 300 to 400 bases. So that is the limitations of the technology. And now if you look at the 16S rRNA gene, which is, as said in the beginning, 1500 bases long, you will see that I also mentioned that 16S rRNA gene has a conserved and hypervariable regions, and I also said that these regions are interspersed with each other. So you can see very nicely there are 9 hypervariable regions which are in between. There are conserved regions. So for us it's a very good opportunity that it is then possible to amplify any single hypervariable region or a combination of two hypervariable regions using making use of conserved regions. So as per your wish, you can either amplify a single hypervariable region V1, V2, V3 individually, or you can amplify them in combination V1 plus V2, V3 plus V4 or V6 to V8, three regions together depending on the read length of the technology which you are using. So it's your choice which hypervariable which you want to amplify, because this is necessitated by the limitations of NGS technologies. NGS technologies, there is no way you can have entire 1500 long molecule to be sequenced at one go. What you can do is you can sequence only shorter regions, and these shorter regions minimum 100 to maximum 400 to 500 bases. So depending on the technology which you are using, what is the read length the technology gives you, based on that read length you can choose a single hypervariable region or a combination of hypervariable region and use suitable primers to amplify these hypervariable regions from your environmental samples, and then use them for direct amplicon sequencing. There you can exploit the benefit of NGS technologies, wherein it can gives you a deeper understanding of microbial community structure. I mean, you will appreciate it better if I explain you a little bit more. See, if you are using capillary sequencing, yes, you can sequence more than 1000 bases region of 16S RNA gene, but then you are resorting to clone library approach, and typically there are two limitations there. One limitation is you have to really get a large number of clones if you want to have a deeper understanding of microbial community structure. So for example, we take a complex system like soil. Soil, we say that soil contains 10,000 different species of bacteria, and if you want to know at least what type of bacteria are present, forget about metagenome, if you even want to know what type of bacteria are present in the soil, then in order to pick up each of the 10,000 different species, minimum number of clones which you want to sequence is 10,000. That is, you are lucky enough that each of the 10,000 clones you are sequencing is a unique clone, which is not the case most of the times. Most of the times, in order to get a better coverage, you have to sequence many more clones. So even now, even if it becomes 10x coverage, which means to say you have to sequence 100,000 clones. So you should have good transformation efficiency, that you are getting that many number of clones, and you should be able to sequence 100,000 clones using capillary sequencing, which is laborious, time consuming and expensive, which can be very easily done in case of NGS, because NGS you get huge number of reads. I mean 300 GB makes several million reads per sample. So it is very possible to go deeper, and advantage from microbiological point of view is that you can pick up a rarest of the rarest phylotype present in your environment. When I say, I'll elaborate it a little more, when we are talking about having 10,000 different species of bacteria in soil, and you take a gram of soil, and let us say that this gram of soil has 1 lakh different bacteria. So 1 lakh bacteria, 10,000 species. So, but it does not mean that these 10,000 species are present in equal number, right? So 1 lakh and 10,000 means 10 each. It is rarely that 10 each are present when you pick up 1 lakh bacteria. There could be some bacteria which are present in 10,000 majority. There could be some which are present in 5, 10, 2 in the given soil sample. And if you just sequence 1 lakh clones, you may miss out this rare minority which is present in lower numbers of 1 or 2 or 5. So that is not possible using capillary sequencing and clone library approach. But when you use NGS, and for a particular sample you are getting, let us say, 2 million reads, then it is possible that in 2 million reads the probability that your rare phylotypes are present in these 2 million reads is more. So that is a reason why these NGS technologies have a significant impact on microbial diversity studies, is because since you get very large number of reads, since you can go deeper in the community structure, the rare phylotypes can also be very easily picked up using NGS technologies. Even if you are using 16S rRNA tagged amplicon sequencing. So, but the disadvantage is that you get shorter reads, and the problem of getting shorter reads you can address by selecting a single hypervariable region or combination of hypervariable regions.
Excuse me, sir.
Yes.
There was a presentation from some company named PacBio.
Yeah.
They mentioned that the read length in their system could go up to 6,000, 10,000, or more than 15,000. And they were saying that they are coming up with a system that where you can get read length up to 30,000.
Right.
So in that case, this problem, the problem of short read lengths, is it possible to, because you are saying that the selection is based upon the efficiency of system, platform,
Right.
So in that case, I guess that problem can be solved, theoretically at least.
Good question. But the problem of, I do not know how much details he has covered about the technology, but basically the problem, yes, the technology gives up to 10 kb read lengths, that is fine, but at least as the situation stands today, the situation in which you can get a 10 kb read length, the error rate is very high.
Yes, sir, he told
Yeah.
that
18% error rate.
Exactly.
So if you want to get a more accurate sequence, then you have to do multiple sequencing, and then your read length becomes shorter. So again, you are basically placed in the same situation. That is one part. And secondly, another reason why one cannot comment on those technology now is that technology is very recently introduced, and there is not much information available in terms of application of this technology to these platforms. So it's a little premature to say how significantly it will have an impact on these studies. And I mean, if you are talking about emerging technologies, there is another technology which is going to come up in the market, and which will probably even supersede this SMRT technology, is nanopore based sequencing, where your sequencer is just as big as your pen drive, and it is very cheap, and it gives you something like how much? 10 GB sequence in 6 hours with a read length of 40 kb. That's what they claim. So, and it is one of the cheapest technology that would be available. So probably that would address some of these issues if that technology is really what it claims to be. But the PacBio technology, I can only say at this moment, is that currently we do not have sufficient information as to how good it is to address these issues, because the technology has been in the market only for last 2 years, and there are very few publications on microbial biodiversity studies using this. There have been some publications on microbial genome sequencing though. But there again, in most cases, exclusively SMRT technology for even microbial genome sequencing, people have not used. Because you go for resequencing using SMRT, again you are losing the cost advantage. So what people do even for microbial genome sequencing for SMRT is that they use a combination. They derive a draft genome using SMRT, and then the finer sequencing on that draft is done using any of the other high throughput technologies, where you get much deeper coverage. That is the standard strategy which people use right now.
There is one more question, sir, about this variable region you mentioned. There is a very common question, and the answer of that question is always incorrect. The only question is asked that which region you will target to identify an organism up to this level.
Yeah, I mean, that answer I'm trying to, I wouldn't say that I'll have a definitive answer, but I'll have some clues to this as we go into my presentation. I'm just preparing the little background for what I was going to say. So right now, we have addressed the issue that we basically use shorter reads. When we use NGS, we get shorter reads. Based on all the available technologies today, we get shorter reads. That's the issue which we are making right now. So if we go a little more details into these hypervariable regions, I have shown here different hypervariable regions and the primers used there and the conserved regions there. Okay, now coming to these 9 hypervariable regions, you can see that the lengths of these hypervariable regions are variable, and the extent of variability in these hypervariable regions is also different. So here the location on the sequence has been plotted against the percentage sequence identity, which means that the extent of conservance of the sequence. And you will find that for example this region V2, the extent of variability is higher because your graph dips down there. You go to region V3 has much higher variability as compared to the regions like V7 or V8. The variability is less. Although these are so-called hypervariable regions, the variability is less. So typically you will see that the V3 region is much more hypervariable. So that gives you kind of guideline as to if you want to capture variability and if you want to get a better assignment of the taxa, which hypervariable region which one should use. So probably this figure itself indicates that V3 you will be able to capture better diversity. See, because if you come to somewhere use this region, then although the sequences are divergent, two different sequences would still may be represented as identical in the V8 region because divergence is less, and they may show as really different sequences in V3 region. So that's what this graph means. So this graph itself indicates that V3 region probably gives you a better capture of diversity. So the major question which everybody asked here is which hypervariable region gives more accurate taxonomic assignments and which primer pair. When you are talking about many primer pair, which primer pair gives better coverage of taxonomic diversity. And what I am going to do is that I am going to share with you a very simple little exercise which we have done to understand this, answer these two questions, and also I will give you 1 or 2 examples of what others have done on the similar lines. So this is again by no means a definitive answer, but it will give you some clues as to how these studies are to be designed. So what in our study we had used the primers pairs which were reported in the literature for various regions. So V3 region there were 3 primer sets, V5 region 2 primer sets, V6 region 3 primer sets, and then V7 region there was only 1 primer set, V1 V2 combined amplification, V2 V3 combined, V3 V4 combined, V4 V5 combined, and V5 V6 combined. So we took these primer pairs and we did a kind of blast and found out which taxa these primers are able to pick up. Then initially we took individual primers, then we took primers in pairs and tried to find out did kind of in-silico PCR and find then okay. What we did initially was we used from the database we extracted all the reference strains or type strains sequences available in the database, and on those sequences then we did in-silico PCR and found out that which primer pair amplifies which taxa preferentially or whether it amplifies all the taxa efficiently. We tried to answer that question on this generated database. So before that we did some exercise. We used E. coli as a reference sequence and okay, this part I have already explained. So we used E. coli as a reference sequence and found out what is the length of the fragment that is amplified using these primers. And as you will see here, in some cases for example, V3 primer pair 3, it at least on E. coli genome it amplified a very short sequence of 14 bases, so that was excluded. For V5, the second primer pair, there was no significant alignment found with E. coli reference sequence, so that was also excluded from the analysis. Then same for primer pairs which amplify two variable regions. This particular primer pair for V1, V2 was found to amplify only a short 19 bases sequence, so that was excluded from the analysis. V4, V5 primer pair was shown to did not show any significant alignment, so that was also excluded from the analysis. And then the analysis was done again. V7, V8 when we used it gave a 308 long fragment, but there was some overlap. So we excluded this also from the analysis, and the remaining pairs were used. So as I said that we used a reference database of all type strengths for the amplification, and then for the so once we extracted the PCR products, we used this in silico generated PCR products to find out taxonomic amplification. So we used the entire type strain sequences database as our experimental database, use these primer pairs to get in silico generated PCR products, and then these in silico generated PCR products were then again remapped to identify their taxonomic affiliations. And remember, we already know the taxonomic affiliations from the database, so we try to find out how accurate taxonomic assignment we get, and for getting this taxonomic assignment we use Green Genes database and also RDP database, and I'll be showing you the comparative analysis of those. So when we use Green Genes database, this is the picture which you see. We looked at the authenticity of the taxonomic assignment at different level of taxonomic rank. And remember, in such metagenomic analysis, you would want to go to as lower taxonomic hierarchy as possible. If you get assignment only up to the phylum, class or even order level, that doesn't serve much purpose. You should be able to go to ideally to the genus level assignment, or at least to the family level assignment, then only you can draw some meaningful conclusions. So what we did, so as expected in this study we found that when you are using a single primer pair, single region, V3 region gives you the better taxonomic assignment, and when you are using the region in single, and when you are using two hypervariable region, a combination of V2, V3 or V3, V4 gives you a better taxonomic resolution up to the genus level. So you can see here it goes almost up to 90%. Combination of V2, V3, V3, 4 goes up to almost 90%. So that gives you better taxonomic assignment. And then again RDP, you have an option of using either 90%. You can decide the percent accuracy that you get in your assignments. So we used two situations, greater than 90% and greater than 100%. So again, situation is much better in RDP when you use at the assignment accuracy of more than 90%. Again you can see that V2, V3 for combined region gives you the highest efficiency of taxonomic assignment. And if you use 100%, again V2, V3 or V3, V4 regions are the ones which give you best taxonomic assignment at 100% assignment accuracy level. So this is as far as the which region to be used for PCR amplification. These results do indicate that probably it is best is a combination of V2, V3 or V3. Ideally V2, V3. If you look at the RDP data, that is a combination which gives you the best taxonomic assignment on the reference data. If you go by the results on reference data, and then if you have to use a single region, if your read lengths are shorter where you have to use a single region, then probably V3 region is a better region to be used for these studies. So that is the conclusion that can be drawn from these studies. What we also did further, hello, yes? Yeah.
And when you consider V3 or V2 regions, then these two regions have variability. But if the other regions are having less variability, then that similarity will affect the resolution power of this V2 and V3. Is it possible?
Sorry,
The dissimilarity and similarity.
correct, yeah.
So if there is more similarity, then dissimilarity will be affected by the similarity size. So in that case, if the V2 and V3 is no doubt it is variable much than the other regions, and the other regions are having less variable, that means similarity will be more in
Yes.
the consensus
So,
sequence.
yeah. So that is probably the reason that it is not able to capture the genus level. It is not going to give you a correct assignment up to the genus level. Since there is a more of conservancy in V7, V8 regions, they are not able to give you accurate genus level taxonomic assignment. That is probably the reason. So that was, when we conclude that V3 region probably gives us the better taxonomic assignment for metagenomic studies, that was not enough. So what we also tried to answer was how good does this region cover all the phyla with equal efficiency, and whether all the ecosystems, how good this region functions with all the ecosystems. And in all the analysis there were some families which were excluded. See, again you should understand that we are working with a known database. The database was generated, was known. So in this known database, some sequences were always excluded, though those could not be assigned to any taxonomic rank in this study. So we also tried to look at which are these groups which could not be assigned properly to any taxonomic rank in this study. So we also looked at these taxa. So when we look at the phylum level assignments, we find that it performs well on the topmost this thing is full length sequences. So full length of course does well, but then V3 region it has for certain taxa it does not give us correct it fails short in certain regions. Some regions are some taxa are better, some taxa perform very badly, and some taxa perform moderately. So a quick look like here it will tell you that it is not so good for proteobacteria, it is again not so good for chlorobia and chlorophyxa, not so good for fusobacteria. So your assignment also depends on which taxa you are working on. For these taxa, it will fail to give you a very accurate taxonomic affiliations. As we go to V5 region, you will see V5 region is not at all good for many regions. It performs reasonably well only for a few groups, otherwise for most of the regions it does not do very well. V6 region again, you see that it is performing quite badly in most of the regions. V7 region, V2, V3 combined, we have been seeing all the time that V2, V3 combined performs very good for most of the taxa. For some of the taxa, the assignment accuracy is less, but it is still well above 85%. And V3, V4 also gives you somewhat similar regions, although for some taxa the assignment level is less than 80%. So again it emphasizes the fact that V3 region is probably the best region, and when you are using combined region, V2, V3 performs better than V3, V4. So that is the conclusion which we can draw from this. So again other regions, V5, V6 you see is not doing very good, it fails for many taxa. V7, V8 region is very bad, not doing, doing very badly for taxa like this chrysogenitis. And so then we also looked at the same data set we used for these ecosystems which were reported in the literature, and we tried to address the question that how good it performs with these ecosystems, and again we came to the conclusion that V3 is proved to be efficient in, I am not presenting all the data, it's a huge data in the form of Excel sheet. So V3 region is efficient in all the ecosystems which we had looked at in this, and V2, V3 is a better primer pair. So from our studies it appears that V2, V3 region, when you are using combined primers, V2, V3 and a single primer, V3 is a better region for such studies. And when we look at the excluded genera, we found that V3, V5, V6, all of them have these genera excluded: Acidobacteria, Actinobacteria, Epiphysi, Bacteroides, Firmicutes, Fusobacteria, but the extent varies. But as you will see, the Proteobacteria are somehow are excluded in by many primers, and V5 regions, maximum number of Proteobacteria are excluded from the analysis. There are some Firmicutes and Acidobacteria also fall within the excluded genera category. So this is as far as what we had done in our studies. There were few other studies where similar results were obtained, and here there was one study where they had generated pyrotags and assigned pyrotags to the reference databases, and where they found they used three different methodologies. We had used only two, RDP and green genes. They had used here best hit, LCA and RDP analysis. And in their studies, they had not used single primers. They had always used combined primers, V1, V12, V21, V34. I mean, they used primers in reverse orientation, and then they found that in their study they found that V1, V2 region probably gives the highest level of taxonomic assignment. But this was a study which was restricted to a biological wastewater treatment. V1, V2 performs relatively better as compared to the other region, but it's just relative. There is not very significant difference. And they had used three functional categories: pathogens, remediators, and active groups. And again they had used three methods for taxonomy affiliation, and they found that under the three different groups and three methods, the correlation is different. In case of pathogens, then when the green is the good correlation with all the three methods, shared by all the three methods. Pink is best hit only, brown is LC only. So depending on the group of organisms which you are studying and the region which you are using, the correlation between different methods used for the taxonomic assignment is different. For the pathogens, there are very few shared taxa, and as seen in green color, whereas best hit and LCA is followed by that in case of pathogens. And so the summary is that based on the kind of group you are looking for and the methodology used for taxonomic assignment, the relative proportion of different forms would vary in different ecosystems. So same way it was done. There was another latest study where a similar approach had been used, and there again they had done a similar study wherein they had again relooked at the taxonomic assignment of different hypervariable region, and they again compared V3 and V6 region, and they found that when you use V3 region at the phylum level, 99% sequences are assigned to a single taxon, 2% sequences were assigned to, 0.04% sequences were assigned to more than 2 phyla, 0% were assigned to 3 or more phyla. So, and for with V6 region, 83% of the sequences could be assigned to a single phylum, 0.17 to 2 phyla, 0.01% to 3 or more phyla, so and so. So in the same way you see that in V3 region 99.11% of the sequences could be assigned to a single phylum, single genus, and 0.07% could only be assigned to more than one genus. So again it looks here that the V3 region performs better than V6 region. So both the study have a common observation that V3 performs better as than V6 in taxonomic affiliation. So this is a summary of the table generated from this study, that comparative taxonomic assignments using full length sequence V3 region and V6 region. So I mean in short one can conclude that V3 region probably is a better region when you are using a single region for taxonomic assignment, and when you want to use a combination, V2 V3 combination gives you the better results. So then I would move on to the impact of such studies on microbial taxonomy, and I'll give you just two features of this. One as an example as to how metagenomic study impact a microbial taxonomy. So this is a very famous study taken up by Craig Venter. He had collected seawater samples from a variety of different locations, and he extracted the metagenomic DNA in the range of 2 to 6 kb, and he generated metagenome sequences using capillary sequencing, and from these sequences he retrieved the sequences for EFG, EFTU, HSP70, REC-K, RPOB, and 16S rRNA, and he used this to study the phylogenetic diversity of that sample. So different groups were identified, alpha bacteria to Crinarchaeota. Different groups were identified using different these 6 phylogenetic markers. And in summary, what he did was, the other part is not important. What is important is that from this study he could discover 1,800 new microbial species or phylotypes just based on the sequences. So there were no culture relatives available, there were nothing grown in the laboratory, but just based on sequences of 6 genes, 1,800 new microbial species could be proposed using this limited exercise. So that was a classical study that was done by Craig Venter. So with the advent of NGS in metagenomic studies, there are many microbial lineages where the information about these lineages is available purely based on gene sequences, whether it is a 16S rRNA gene sequence or some other gene, and this is reflected in the this next domain bacteria, phylogenetic tree of domain bacteria, where you will see that the blue colored branches are the ones where at least one cultured representative is available. So these are the branches where minimum one cultured representative is available. The red colored branches are the one where there are no cultured representative is available. All the information of about these lineages is coming from the metagenomic sequences. And there are, I'll just quickly mention this, there are green dots outside the these red circles. So these green dots mean that for these lineages at least one reference genome sequence was obtained using single cell genomics. So I am not going to what is single cell genomics, but here you do not grow the organism in a laboratory, but you just try to isolate a pure single cell and do the metagenome sequence on that. So for these lineages at least one genome sequence was obtained using single cell genomics. So this is where the microbial taxonomy stands today with the impact of metagenomic studies. And okay, so then I'll quickly go through some other part which I had mentioned. So if you want to have a break here and ask some questions, I'll answer the questions on the first part of my talk, and the next 5 to 10 minutes I'll go through the other part which I want to go through quickly. Yes.
Currently, the cutoff percentage of similarity in 16S is 97 or 99%?
When you are talking about, there is a proposal, when you are talking about a complete 1.5 kb sequence, it is recommended that 98.5% is a better value than 97. But again, it varies from genus to genus. For some genera, it could be more than 99% also.
So if you consider only B2 and B3 region, then the percentage will be different.
When you go for, see what I said was for 98.5% was for the complete genome sequence, 1.5 kb genome sequence. When you go for hypervariable regions, when you do OTO assignment, you use different cutoff values. So sometimes that cutoff values could be even at the level of 99%.
Okay, so there is no standard for these variable regions.
There is no recommended, see when you use these online softwares, there are certain default values. So you do the analysis using the default values, but then if you do not get a satisfactory phylogenetic picture, then you have to modify the default values and set new values and see what kind of picture you get, and then you interpret your results with that cutoff value.
Actually, sir, because the basis of cutoff value is on our established fact that these are two different strains, which is practically seen by a microbiologist, and then without knowing its background of morphology and biochemical activities, then somehow we can set a strict percentage of similarity dissimilarity to assigned species in a metagenomics.
See, no. I'm not getting your question now. Setting cutoff value in metagenomic studies is one issue. Setting cutoff values for your isolated microbes is a separate issue. See, metagenomic studies, when you do, you have no idea about the strain level differences. So there you really cannot compare the, because you don't have the isolated organisms in your hand. So there you have to go whatever you see at the overall community structure level. When it comes to discussing the cutoff values of isolates, then it's a different situation. You look at it in a different way, and that's why I said that when you are talking about isolates, the cutoff value largely depend on the genus you are working at. For example, the genera like Pseudomonas, Bacillus, Aeromonas, to a certain extent Streptococci, there the even 99.5% cutoff value is not sufficient, because many well-established species have homology values up to 99.9%. I mean, there is a classical example in case of Aeromonas where two well-established species have only single base difference in their 16S rRNA sequence. So there it would depend on which genus you are talking about. The cutoff value, even 99.9% 16S rRNA sequence similarity may be giving you a different species provided other values are different and other things match. So at that level we talk about cutoff value in a different way, and when we are talking about setting of a cutoff value in OTO assignment in metagenomic data, there you can there the cutoff value has a different meaning.
Sir, as far as metagenomics and single cell genomics is concerned,
Right.
so whatever you have talked about single cell genomics, that
Right.
can be done from the same sample what is collected for the metagenomics, or it may be concerned with the unculturable diversity of particular organism, or
See, I mean, just to give you just one example, I mean, I don't intend to go into the details. These two things are correlated basically. Metagenomics and single cell genomics are correlated. Just to give you one very simple example for the sake of understanding. For example, you have taken a lake water sample and you have carried out a metagenomic analysis of lake water sample, whether 16S rRNA or whether whole metagenomics, and from your data you feel that one particular phylotype is important and you want to know more about that phylotype. So at that stage what you can do is that you can develop fluorescent tag for using the DNA sequence. You can develop a fluorescently labeled hybridization probe for that particular phylotype. Then you stain your sample with that fluorescent tag and then do a fax sorting. So during the FACS sorting, then the bacterial cell carrying that fluorescent tag will be sorted, right? And then is your single cell genomics. So you have sorted a specific phylotype for which you want to know more, and this is an unculturable phylotype. So there then you go for single cell genomics and get the entire genome sequence of that isolate and see where it fits into your overall metagenomic picture. And this need not be a single, we just said a single phylotype. But if you find 10 different phylotypes are more important, you develop 10 different fluorescent tags and do single cell genomics of 10 different phylotypes to get the entire community structure picture for that particular sample.
Sir, I have one.
Yes.
When we check with the combination of primers,
Yeah.
V2, V3 what are the combinations, whether the classification is based on the OTU based or sequence based?
No, see in the study which I presented, there we had used the all type strain sequences, and we had reassigned the in silico generated PCR products. We had tried to reassign to the reference 16S RNA sequences of reference strains.
Reference strain. So
Yeah,
that
so there was nothing like OTU generation or anything there.
So sequence similarity.
Yeah. Any other question? Okay, so then I'll quickly go through some other interesting applications of metagenomics. So, metagenomics also has a lot of industrial applications. Besides studying human microbiome and ruminant microbiome and other microbiomes, it has a huge application in industries. And if you look at this figure, the figure A is the enzymes that are available for biotransformation, which are currently being used in various industries, and you will find there are hydrolysis, lyase, isomerases, ligases, oxidoreductases and transferases, and metagenome has a important source to get novel biocatalysts. And right hand side you will see that the available metagenome sequences has huge repertoires of oxidoreductases and transferases, and transferases are something which we are running short in the currently available industrial enzymes repertoire. So we may have a good novel transferases from metagenomes, and then same is the case to a certain extent with lyases and isomerases also, because isomerases also we have very few isomerases currently available, but metagenome so far whatever we have found has a good number of novel isomerases. So it would the industries would definitely be looking for novel enzymes in metagenome, and there is a novel antibiotics which have already been reported from metagenome. So it has huge industrial applications, and this is a long list of enzymes which have been novel enzymes which have been discovered in metagenome. So you will find xylarases, proteases, deoxygenases, amylases. A large number of novel enzymes have been reported from metagenome, and all this is not just fancy, but at least there is one industry which is actively promoting the enzymes that have been discovered from the metagenome with novel properties. So in their basket they have enzymes, they have phytases, they have amylases, they have glucoamylases, and several other enzymes they have in their basket, which have basically been generated from metagenomes, where they have process optimized and overexpressed these enzymes, and these are available commercially. So these are the other enzymes that are available, and then a quick look at what we are doing. So we are looking at human microbiome, various aspects: mother-infant microbiota, age-related differences, genome sequencing, diseases, celiac, diabetes, and IBD. So I'll just give you a bird's eye view of that. We had already published this work where we had looked at families, we had looked at the succession of microbiota, and there, so we had already we had looked at the succession of microbial communities in two families, and we had also done some isolation of anaerobic bacteria, and what we found was that there were large number of, from a single study there were large number of potentially novel isolates based on 16S RNA gene sequence. So we had again I'll come back to that in a moment, that few of them have already been described as a novel taxa, and we have also done genome sequencing of these isolates. We had also studied the effect of mode of delivery, but in this case there was not much difference found in based on the mode of delivery, but that was because here the samples were collected from teenagers, and by the age of 3 to 4 years the gut flora gets stabilized in the individual. So that's the reason we have not been able to capture the differences between cesarean section and vaginally born babies here. But in the same study, when we compared this population with that in the Finland same age population, we found that the Indian teens, they form a green color, they form a very distant group as compared to Finnish teens. So this is there could be various factors contributing to that, genetic factors, environmental factors, and regional differences. We also, this is a latest study, we had also generated tag amplicon sequencing data for a large number of individuals from two different parts of India, and we compared that with global population. And here you can see the representative picture of one American study where we have compared our data with that, and we find you can see very clearly that they form two distinct groups. So Indian population, you can see it in the right hand part of our of this figure, and left hand side you can see American individuals. So they indeed form a distinct group. We have also compared this with population from other part of Asia like Korea, China and Japan, and we find that it is indeed very different from rest of the world. Then we had also done one study wherein we had looked at a disease called as celiac disease, which is basically a gluten intolerance. This was a follow-up study where the patients with celiac disease were given gluten-free diet, and their gut microbial population was followed, and you can see that at this extreme, as the disease patient before the start of gluten-free diet, and as they are put on gluten-free diet, their gut microflora starts moving more towards normal. So at the extreme, when they are diagnosed of the disease, they have very different microflora. And we have also done genome sequencing of some of those isolates and our gut isolates, and this is, I can summarize these results for one of the organisms, that red triangle is the carbohydrate metabolizing enzymes of human origin, and here the green square, these green circles are the ones, two megaspira isolates which we have generated from human gut, and it shows the carbohydrate metabolizing enzymes from these two isolates. And you can see that this part, this rightmost corner, you can see that these are the carbohydrate metabolizing enzymes which are exclusively produced by these two isolates, which are very different from those produced by human gut. So, one can conclude that these bacteria contribute a unique repertoire of carbohydrate metabolizing enzymes in our gut. So, this was one example, and then another very interesting example of metagenomic studies which we had done was, I will not again go into the details, but just to tell you how functional metagenomics also can give you some interesting information. This was a study done on a effluent treatment plant in Andhra Pradesh, which treats the waste from antibiotic industries. So we had generated a functional metagenomic library. From this we had extracted the metagenomic DNA, and it was cloned in a vector. And since we are looking for antibiotic resistance genes, the clones were straight away plated on antibiotic containing media, with the idea that only those clones which have antibiotic resistant genes would be expressed, and then we PCR amplified and sequenced the antibiotic resistant genes to find out what kind of novel genes are present. So this is a vector which was used for the cloning, and the antibiotics that were used are carbanicillin, ampicillin, ciprofloxacin, tetracycline, gentamicin, amikacin, azithromycin, and sulfamethoxazole. So these are the antibiotics which these were used in this study, and then we could indeed get this is the functional distribution of these antibiotic resistant genes at different levels of effluent treatment plant. Again, I will not discuss this in details, but eventually what we found was that we detected resistance genes for all these group of antibiotics which we had studied. And not only that, at the end of the study, 20 out of 26 instances were positively conferring antibiotic resistance. I mean, this was the transfer study. But finally, what we had also discovered there, 38% clones had inserts with putative new resistance genes. So these remaining clones had the, when we sequenced the clones, the remaining clones had the known antibiotic resistance genes which were already known, but 216, which is 38% clones had potential new antibiotic resistance genes. So the point is that the antibiotic treatment plants could also serve as a potential reservoir for new antibiotic resistance genes which can be recovered in metagenomics studies. So this I thought was one interesting application of functional metagenomics, so I thought of sharing this with you. Thank you. Any more questions? Sure.
During the introduction, your work is also on insect gut microbiome.
Right, yeah.
Can you throw some light on that?
Yeah, see, insect gut microbiome, we had initially started the work on mosquitoes, and still we continue to do it in a smaller way. What we had looked at is that role of insect gut microbiota in development of mosquitoes and their capacity to transmit the diseases. So we had done the in human they do the studies right from the birth up to this thing. So we had looked at the microflora at different developmental stages in eggs and larvae, and we had looked at role of microbes in the capacity to transmit dengue, for example. That was a study which we had done. And then we do a lot of work on endosymbiotic Wolbachia in insects.
And what is the status regarding the Wolbachia and the relevance with the nematode Onchocerca?
I have not followed the latest this thing in Onchocerca, but from what I know is that there are two things which I know about Onchocerca and Wolbachia. Is that one is that inflammatory reaction of the Onchocerca is basically because of the polysaccharide of Wolbachia, and tetracycline treatment to get rid of Wolbachia also reduces the infectivity of Onchocerca. And that is the information which I have right now.
Means nowadays scientists are telling that instead of Onchocerca volvulus or other parasite, the river blindness in the human being, that is caused by the Wolbachia.
Yeah, I mean that's what I meant to say is that the pathogenicity of the Onchocerca is probably because of the Wolbachia, and this is something which is not unique only to Onchocerca and Wolbachia, but nowadays there is a more and more evidence that many things attributed, many properties attributed to many organisms are primarily because of the bacteria associated with them and not because of that organism per se. So one example is, as you said, that as an inflammatory reaction in case of Onchocerca is actually because of the polysaccharide antigen of Wolbachia. So that is one example. And another example, it has also been shown in many publications that sponges produce very interesting bioactive compounds. And now the research shows that majority of these bioactive compounds actually owe their origin to bacteria associated with sponges and not sponges per se. So there are many such things that are coming up where the actual causative agent is microbes associated with the organism and not the organism per se.
Sir,
Yes.
so in the 16S DNA sequence based approaches, sometimes we come across unidentified, unculturable bacteria.
Right.
So is there any way to characterize these bacteria further? Any technique which can be used for further characterization?
See, I mean, this is when you are talking about a situation where you have a isolate and the sequence shows uncultured bacteria is, yeah. So when you have a isolate for which you have a 16S rRNA gene sequence and it shows you a hit as uncultured isolate from XYZ environment, then the first thing to do would be to refine your analysis and look for the closest cultured relative that you get. So from that then you can get some information as to what kind of organism it is, and then from there you can do a further, first of all, then once you have that closest culture representation, you can do some comparison of properties of your organism and that organism, and then how good they are matching, you can take your study further.
So it can be a good potential project.
Yeah, it is a good potential project, and many times it could definitely lead to a description of a novel taxon. That much I can tell you.
Sir, as she mentioned, that is it possible that if we are having a culture that is growing well and still it is showing homology to uncultured bacteria after,
Right.
7%
so how is it possible in that way?
See, that is more to do with the analysis which you are doing. Typically, if you do this analysis in a NCBI database, you will end up with such observation, because NCBI database, when I do a 16S RNA based metagenomic study, I submit my sequences as uncultured sequence from uncultured clones, right? I have generated 1,000 sequences in my 16S rRNA clone based study, right? When I submit them to NCBI database, when I describe the sequence, I describe it as an uncultured clone XYZ from this ecosystem. Now when you have an isolate whose sequence matches with my clone sequence, it would give you identity as uncultured clone. So if you refine your study and use some other curated database, or even NCBI analysis itself, you go down and see which is the closest cultured representative, that will give you the more appropriate picture of status of identity of your organism. If you go to a database like RDP, you can do it in a better way. RDP gives you option that whether you want to compare your sequence with isolates or uncultured clones, both. So you give option of only isolates. That will tell you what is the cultured organism it is closer to.
That uncultured is necessarily uncultured?
Not necessarily, that's what I said, that it is because of the entry in the NCBI database primarily. Yeah.
So there is one question, when we do a metagenomic analysis, we normally get the sequences of many organisms that are not cultured.
Right.
But how can you be sure that we have not missed out something? Like say, on the site we get 3,000 organisms, or the 3,000 sequences. How do we know that it's not 30,000?
Yeah, see, before you do this metagenomic analysis, depending on the complexity of your ecosystem, you need to decide the depth of sequencing what you want. So, so for that you need to know, so have some preliminary advantage, preliminary knowledge of your ecosystem. Obviously, a less diverse ecosystem like insect gut. I mean insect gut, I do not mean to say a very complex insect, but a more, what should I say, more specialized insect, like the insect surviving on some toxic compounds or something like that. Or let us say, forget about insect, let us take a ecosystem like hot water spring. So hot water spring would have much less diversity. By virtue of extreme temperature, one would expect much less diversity in hot water spring than a normal lake water. So Of course, lake water itself is less diverse by virtue of it being a oligotrophic environment, but still it will be more diverse than a hot water spring. So your ecosystem, which ecosystem you are working on, would first decide the extent of coverage you want. Ecosystem with much less diversity, you need not have a very large metagenome sequence to get a complete coverage. You could do with a less coverage. But a complex ecosystem like soil, marine sediment, human gut, or any mammal gut would require much higher coverage to get a complete metagenomic picture. So your complexity of your ecosystem would decide how much coverage you want. Larger the complexity, much deeper coverage you need to understand the metagenomics, and it would also depend on the kind of question you are asking. For example, if you are doing a comparative study on effect of diet on a particular organism, so you are giving 5 different diets and looking at the effect on the microbial ecosystem. In that situation, probably you do not have to go much deeper in the diversity, because you want to compare between 5 different situations. So you just want a comparative analysis how the microbial communities are shifted, and you can do it at a upper scale rather than going much deeper. But if you want to really study the functional aspects of a microbial community structure in an ecosystem, then you want to go much deeper in the metagenomic studies. So there you require a deeper. Thank you.
Thank you very much, sir, for such a nice talk. Now I request Ram sir to please come and felicitate Momento to Shouche sir as a token of love and appreciation.